Okay, welcome to the VacCAP webinar series. My name is Amaya Tucha and I'm a member of the VacCAP team and I will be today's webinar moderator. Today we'll be discussing developing a new methodology for evaluating texture in cranberry fruit with Hector Lopez-Moreno. Hector is a PhD student in the plant breeding and plant genetic program at UW-Madison, working with Dr. Juan Salapa. His research project focuses on cranberry fruit quality traits such as firmness, external appearance, and internal structure, and includes the development of phenotyping methods, genetic mapping, and multi-omics approaches. We're very excited to have Hector talk about his current research. I'm going to ask everybody please to stay muted and with their cameras off. If you have questions, please feel free to put them in the chat box, and we will ask those questions at the end of Hector's presentation. So I'm going to let Hector take it away from this point on.
Can you see my screen now?
It looks perfect.
Okay. Hi, everyone. Thank you, Dr. Maya, for the nice introduction. Thank you, everyone, for joining for today's webinar. So the topic for today is developing a new methodology for evaluating texture in cranberry fruits. And I will basically discuss two papers that we published for this as part of this research project. So let's get started. So, cranberry is a crop native to North America that is popular for the nutraceutical potentials. And this crop has become so important that the production has doubled since 1990, with North America being the largest producer in the world, accounting for about 96% of the total production. So, this crop is highly industrialized because approximately 95 to 97% of the total production is destined for processing industry, and only around 3 to 5% is used for fresh consumption. Historically, the cranberry market has been dominated by the cranberry juice. That is, this high dependence has led to the industry to face challenges such as price fluctuations and overproductions. These challenges has been addressed in different ways, such as regulating production volumes and more recently promoting the diversification of processed products to reduce dependence on a single product. So it is evidence that the priorities in the cranberry industry are changing, and the breeding programs are not an exception. So to determine the new priority traits that should be considered for the future cranberry varieties, Karina Gallardo and college conducted a survey at cranberry stakeholders meetings across the US and Canada. And this study included a response from more than 500 individuals, including growers, nurseries, processors, breeders. So this provides a really good representation of the community. In summary, the results show that for the priority food quality traits, the priority traits are firmness, fruit size, and antioxidant content. In particular, fruit firmness is a priority because it determines the fruit performance under storage condition and is a crucial factor for increasing the production of sweet and dry cranberries, which is currently the most profitable processed product. So in this context, we must address some limitations. For example, the current varieties don't fully meet the firmness requirements, especially for those emerging processed products. And, and we essentially know nothing about the genetic basis of these complex traits, which could be useful for more efficient breeding processes. Last but not least, we don't even have a standardized methodology for texture evaluation. Also, there has been some reports on texture evaluation. Both academia and industry has been using completely different methods, making direct comparison of the measurement difficult and slowing down the progress in research and breeding. So to address these limitations, one of the objectives for our research is to establish a reliable method for measuring cranberry fruit firmness. To achieve this, we follow basically four steps. First, we identify potential methods that could work for cranberry based on the literature from other species, such as blueberry, strawberry, apple, and other berry crops. Next, we consider the selected methods. Considering the selected methods, we analyze several factors that have been reported to affect the performance of the texture measurements, such as storage time, the temperature of the fruit, and different settings that can be adjusted in the texture analyzer. Then, based on the optimal conditions that we previously defined, we compare the methods under harvest and post-harvest conditions, and finally, we propose a set of traits able to differentiate cultivars based on different texture components as part of the standardized methodology. So the first 2 points were addressed in the first paper, and the third and the fourth points were included in the second publication. And now I will discuss these two publications in more detail. So the title for the first publication was A Survey of Key Method, Traits, Parameters, and Conditions for Measuring Texture in Cranberry. Here is a summary of the resources and tests that were included in these first publications. 5 different methods were evaluated with a total of 47 traits, and the methods included were double compression, single compression, puncture, blade, and Cranberry chisel. These methods were tested considering cultivars that span the full firmness spectrum of cranberry to ensure a good representation. In total, we conducted 7 different tests related to factors that can affect the texture evaluation. For example, the determination of the optimal sample size, the correlation between the fruit size and firmness, the effect of the storage time, the effect of the fruit temperature, and the optimization of the speed straight combination, and the effect of the probe diameter for the puncture test. Uh, to summarize the main results of these papers, here are some key considerations for texture evaluation. The first is that 30 fruits are required to obtain accurate results for all the methods. Another consideration is that firmness reduction is genotype dependent, which has practical implications, especially when working with different genotypes. We also reported that firmness reduction remains mostly stable until the first month, where it decreased only around 10% on average. The third point is that some conventional traits, such as maximum force, which is widely used, might provide bias estimations influenced by the fruit size and shape. In contrast, more sophisticated traits like apparent modulus of elasticity, maximum contact pressure, account for size and shape in their calculations and could provide more accurate evaluations. The final point is that the combination of the speed and strength determines the ability to differentiate between cultivars and is specific to each trait. For example, a trait that like maximum force that can effectively distinguish between cultivars with a specific speed and strength combination, but this might lose that effectiveness if a different speed and strength combination is used. So that was a brief summary of the first publication, which provided essential information for conducting these second studies that is titled Multiparametric Cranberry Fruit Textural Trait Development for Harvest and Post-Harvest Evaluation in Representative Cultivars. Similarly, here we have a summary of the study. In this case, we use 5 methods with a total of 22 traits, and I would like to point out that we use fewer than half the traits that compared to the first publications because we discarded the double compression method, which was very similar to the single compression test. Instead, we decided to include another compression variance that fractured the fruit. So In this case, we use 5 methods, including single compression at 10% of the strength, uh, single compression with 50% of the strength, puncture, blade, and Cranberry shear cell. We use 10 different cultivars that cover the full range of firmness, and in total, we conducted 6 different tests in both harvest and post-harvest conditions. And next, I will explain some of the most relevant tests and findings from this publication. So first, we perform a general comparison of the 22 traits from the 5 methods at harvest to select the most effective methods. For this, a principal component analysis was implemented as a dimensional reduction and variable selection method. And as you can see, here we have a lot of information in these two plots, and I will explain this in more detail. So the first thing to note is that these traits were able to capture a large percentage of variance, around 84%, which is very high. This is a very good result that demonstrate these traits can capture the texture characteristics that differentiate Cranberry genotypes. Now let's focus on how the cultivars are differentiated according to this analysis. We can observe that there are 3 main groups that are well differentiated. The genotypes with firm fruit, highlighted in red, which included genotypes such as M34, DP17, DP14, and granule red. Then we have genotypes with intermediate firmness like Stevens, Sundance, and GH1. And finally, we have the soft cultivars with BG, V95, and Yellow Bell. Now, let's take a look at the projection of the traits in the quadrants of this plot. Um, One of the main observations is that the traits that can be grouped, the traits can be grouped in 2 main categories. Destructive, that is highlighted in red, and not destructive, that is highlighted in blue. Sorry. For example, for the compression traits at 10% of the strain, which don't damage the fruit because they only slightly compress it, are grouped together and aligned with the PCA1. And we can see that these traits are here. And this PCA1 is the one that captures the largest percentage of variance. On the other hand, we have the cluster of the destructive traits from the other method that is over here. And these traits damage the pericarp in some way, either through puncture or fracture. So these destructive traits primarily align with the principal component 2, which is less relevant than the principal component 1. This suggests that the cranberry texture can be better captured using less invasive methods, such as the compression test with 10% of the strength. Another observation that we can make here is the redundancy of the variables. The protection of the of the variables is also an indication of the correlation between the traits. Um, So the more similar the projection of the variables, the more possibly correlated those traits are, and vice versa. Here we can observe that the compression traits are mostly redundant because they are clustering together and providing similar information. But in contrast, the other traits are less redundant and might capture more diverse texture properties. This is part of the work that these dimensional reduction analysis do, so we can focus on the most informative variables. So another important piece of information here that we need to consider is the contribution of the traits to the variance explained by the PCA 1 and 2 in this case, and I will explain that below. Mm. So this is a bar plot that shows the traits ordered according to the contribution to the PCA1 and 2, with the highest contribution at the bottom and the lowest contribution at the top. It is clear that the compression and the puncture are the most relevant uh, methods based on this criterion. Considering all the points mentioned for these results, for the results of the analysis, we decided to focus the next steps exclusively on these two methods, puncture and compression, as they were the most promising. So next, the next step was to evaluate the compression and puncture under storage conditions. And during storage, the firmness decreased during because of different biological processes. So we are confident that we will see changes over time. Therefore, this is an opportunity to select those traits that best detect the difference in this scenario. So first, here are the results for the compression 10% of the strength. For this test, we also use a principal component analysis, and in this case, in addition to the genotype represented by colors, we have the variable storage days with four different points: 31, 86, 163, and 288, that are indicated by the size of the points, as shown in this legend. This way, each point represents the sample for a specific genotype for a specific period of time. Now Let's break down these results. So the first observation I would like to make is that the high we have here a high percentage of variation explained only by the PCA1 is about 98%, which is pretty high. And as you can see, all of the traits are aligned with this principal component. These traits are also doing a really good job in differentiating between the different time points for every genotype. And as we can see in the example highlighted for the BG genotype over time. For example, here we have the last storage date, the second, the third, and the first one. So all the storage time for this genotype are very well separated. The projection, the projection of these variables indicates that the traits are mostly redundant as they are clustering closely together. As we can see in those two clusters that are very close. However, there are two small groups. The upper cluster, which includes the traits, and those that consider the size and the shape of the fruits, such as apparent modulus of elasticity and maximum contact pressure, slightly separate from the rest of the traits, which are grouped in the lower cluster, in this cluster. So even though these two groups or traits differ only slightly, it is crucial to consider this distinction, especially when these measurements will be used for analysis where maximizing the statistical power is crucial. This is particularly true, for example, in genetic mapping studies where such difference could determine whether we find some specific regions or in the genome or not. As mentioned earlier, the contribution to the variance of the principal components is important for selecting variables. However, in this case, it is not that relevant since all of the traits have a really high contribution and the contribution is very similar, as we can see in this bar plot. For that reason, we decided to select the three highlighted traits, because they covered the two cluster traits that we identified previously, and therefore maximum force, parent modulus elasticity, and maximum contact pressure were chosen for further testing that I will explain in more detail later. But now let's gonna check the results for the puncture test. So for the puncture test, we followed the same approach. evaluating genotypes at different storage time, but the results were notably different. To begin, the variance explained by the principal components are more distributed. Um, However, the first two principal components still account for approximately 97% of the variance, which is also a really high percentage. These traits are also able to differentiate between the time points for every genotype, as we can see in this example for the yellow bell genotypes. The different points are indicated by the red arrows. Another contrasting results of this method is that the traits are not as redundant in contrast to the compression traits. As we can see in this biplot, the different traits are more spread out across the quadrants. Now, according to the contribution in the panel B, in this bar plot, we can see that the traits had similar total contribution to the PCAs. However, they mainly differ in terms of their contribution to each individual principal component. Taking all these observations into account, we decided to select the traits work, that is over here, and the trait deformational rupture, that is over here, and also over here, since they provide a good representation of the results from this test. Then we proceeded to conduct additional evaluations for the selected traits for the compression and the puncture trait test. Sorry. One this evaluation was to monitor the texture dynamics during storage to analyze the trend of these traits under these conditions and potentially demonstrate their use for identifying cultivar with good performance. These are the profiles for the 10 cultivars during storage for the 5 selected traits. In all the traits, for both the compression and the puncture methods, and the order of the cultivar profiles is largely consistent. This means that in almost all the cases, the order of the cultivar showed in the left is very similar for all the traits. For example, we have that for most of the traits, M34 and DP17 are the firmest cultivars, and yellow bell is the softest. These profiles also shows us that some cultivars such as M34, that is coloring gray, and DP17, that is coloring green, tolerate storage quite very well, even having higher firmness values at the end of the storage time, that is over here, compared to the values at the initial test with some soft cultivars like yellow bell, B95, and BG. So this demonstrates that there is a high variability in the cultivar response during the storage. Finally, we conducted a preliminary test for with fruit that were previously frozen to try to mimic some of the industry storage conditions and identify traits with potential to be used for this purpose. So here are some key points to summarize this preliminary test. The fruit were frozen at minus 20 Celsius for 60 days and were subsequently evaluated once thawed. Then, as shown in the box plot, the measurements were compared between the thawed fruit and the frozen and the values from the fresh fruit to see if there was significant difference within cultivars, and also to see if there was correlation between the fresh values and the frozen and the values from the frozen fruits. So the results shows that the texture values decrease drastically. For example, up to 90% for maximum force and up to 270% for deformational rupture. The only trait that showed a significant correlation was maximum force. which indicates that this trait has potential for use under these conditions. This accuracy will likely improve if specific parameters for speed and strength are defined for these fruits that were previously frozen. So the conclusions are that the compression and the puncture traits are good discriminant of cultivars at harvest, storage, and partially maintain the performance with that fruit. The high effectiveness of the compression traits under different conditions demonstrate the relevance of the structure and the flesh in determining the texture of the cranberry fruits, as these are the characteristics primarily captured by this method. Some of the traits of our flat prop puncture method show good performance and are of special interest to capture the, the effect of the skin. However, we anticipate that by implementing needle prop puncture, this result could be improved. So the complementary use of this method is recommended to achieve a more comprehensive evaluation. From the original 47 traits, we only selected 5 different method traits, sorry, from the compression and the puncture test. That are the maximum force, maximum contact pressure, apparent mode of elasticity, and for the puncture test, we propose work and deformational rupture. Thank you. Let me know if you have any questions.
Thank you, Hector. That was really interesting. Do we have any questions for Hector? And we are a small group, so if you want to unmute yourself and ask a question, I think that's fine. If you don't want to do that, you can just type it on the chat, and I can just go ahead and read it out loud.
Maya, this is Jim. I have a question for Hector.
Go ahead.
Yeah. Okay.
Hector, do you know how any of these texture readings that you took relate to processing for SDCs? Are there sort of ideal parameters for making SDCs?
We don't have direct comparisons. However, the more I could say the more related information to that will be that for example, the maximum compression trait that is able to capture the firmness after the fruit were frozen is a good indication that could probably provide good results for that to predict that the performance of the cultivars for the production of sweet and dry cranberries, because as you probably know that the fruits are usually frozen before they are processed, so that could probably would be a good indication. But until now, we don't have a direct comparison, but that is one of the tests that we are planning to do.
Thanks.
Mhm.
One thing that is worth mentioning is that the fruit the fruit storage seems to indicate that fruit, when they're stored as they get older, the firmness is maintained and in some level. And I think what happens is when we take fruit that is frozen, there's a similar loss of texture when things are frozen. So I think that this is potentially applicable to SDC production because of the way that the fruit loses firmness during storage. We did some testing even with frozen fruit that Hector didn't cover here, but I think the level of firmness by firm varieties remains, and that could be useful. So those are tests that we are doing in VacCAP 2. What is the relationship of firmness to the actual processing with Dr. Diquera? But I think there is good indication that this could be relevant to SDC production. Thank you.
Gotcha. And I'm also curious, Hector or Juan, are there any physical characteristics that you find correlated with the texture readings, such as thickness of the skin or thickness of the pulp or the size of the locules, anything like that?
So far, we noticed that there is a correlation between the fruit size and the firmness, and that's why one of the reasons why we try different texture traits, because some of them are more influenced by the size of the fruit, but others take into account that information. Uh, about the internal structure, I remember. Don't remember exactly right now the correlation, but I remember that there was a correlation between some traits of the internal structure with firmness, which I think has been also reported in other crops that have a similar internal structure, like tomato that has locules.
Yeah, and I think also that we have a third paper that is going to be the internal structure. And then, so right now, everything that we've been doing is firmness, but now we're going to bring in internal structure. And I think, what is the other group of, in external appearance? And so now, right now, we're only analyzing weight and firmness, but external appearance has a size component, and internal structure has a fleshy component. So I think all of that is going to be analyzed in the third paper that Hector is working on.
Sounds good. Thank you.
Hey, hi.
More questions?
Yeah, hi, Hector. This is Rod Saris. Can you hear me?
Yeah,
So,
sure.
yeah, good. Yeah, I was just wondering, I missed the first few minutes. Sorry about that. But how did you, I guess, when did you harvest the fruit? And have you done any firmness or texture analyses of the maturation time? Because that might make a huge difference in how, the texture firmness of the fruit of when in its maturation period that you actually tested them.
Yeah. Well, the fruit that were evaluated here were evaluated at the maturity stage. Actually, the genotypes were harvested on different dates, because a previous report showed that these traits, the same set of traits that we use here, have different peak for the firmness values. So in different dates, I don't remember exactly the dates, but some of them for like, I don't, just to say something, for the middle of September or at the end. So based on that consideration, we harvested the fruit in some specific dates in order to evaluate these genotypes like in the same physiological state.
Okay, so just to understand, so based on historic research, you tried to pick the maximum firmness date for harvest
Yeah.
for that variety? Okay.
Yeah. Mm-hmm.
Okay.
Well,
Thanks.
basically trying to capture the physiological maturity.
Mm-hmm. Yep.
So can I ask a question to clarify that?
Oh, yeah.
Yes.
What did you base your ripeness evaluations on?
The ripeness?
Yeah, you said you harvested based on ripeness.
Yeah. There's a paper that was published by a former student in our lab that basically evaluated a lot of traits, including internal structure, texture, and also fruit color. Based on the different traits, they provide information about when is the when the different cultivars reach the physiological maturity, to say in some way that, for example, reach the maximum fruit size, or that the fruit start changing the color, or when the firmness reach the peak of the values. So we basically use that information to harvest the fruit when they reach the highest value for firmness, in order to try to have the same condition for all of them.
Okay, thanks. I think that clarifies.
We have a question in the chat from Pedro. Says, what for quality components do you think are making a specific genotype lose more or less firmness during storage? Example, bigger fruit might lose weight faster, thus affects firmness.
Storage. Well, I think there is different factors. One could be also the size of the fruit, for example. I remember there is a paper about blueberry, if I remember correctly, that they check the difference in the cell wall disintegrations during storage, and that provide difference between cultivars. That could be also something that could be differentiated between these cranberry cultivars. That could have like a genetic basis for that. Yeah. Also, the content of the some other compounds like rhamnose or fiber or pectin could be involved in that.
So jump in with one more. You mentioned the possible correlation between size and firmness. Yeah, I know yellow bell's probably your smallest fruit in this cultivar sample, and DP17 is a quite large fruit, if I remember right. How was that correlation? Was it pretty strong? Bigger fruit always were firmer than smaller fruit?
For those genotypes,
Yes, yeah.
actually, yellow bell don't necessarily have the smallest fruit.
Huh.
Is interesting because it's the softest, but not necessarily have the smallest fruits. But actually, the cultivars that we use here have um, a kind of variation on in the fruit size. They are kind of uniform, but overall, they are like kind of similar, I would say.
Okay, so the cultivars that you chose, they're all mostly in the middle to middle large range. Is that what you're saying? Versus a few outliers, maybe. I know there are some varieties that are huge. I don't know if you had it, like Pilgrim King, I think, is a very large fruit. Yeah, so the correlation between size and firmness isn't that important, it sounds like.
Mhm. Yeah, and actually, well, in the first paper, I just covered a few points, but actually, in that paper also we talk about a specific test where we basically create choose fruits with a certain size, and we created three different sets, and then we run the test in texture test in that set of fruits. So in order to analyze in more detail the correlation between fruits and size and firmness, um, some of the traits are influenced by size, but not all of them. But overall, all of them has some degree of are affected in some degree by the size, but others are more affected than other traits. For example, the maximum contact pressure, the, the calculation for that trait, the formula, you have to provide the diameter of the fruit, and this calculation was designed specifically for convex shape objects. So that's trait actually have a lower correlation compared to other more simple traits like maximum force. I don't know if um, I was your question. Sorry,
No, I think that was good. Yeah,
mm,
sounds like it's not the terribly strong correlations, and you looked into that within a variety as well.
mhm.
Okay, thanks.
Mhm.
I have one question for Hector.
Oh yeah.
So Hector, do you think you have any other data that could along the lines of what you were saying about the fruit being convex? That's what I also was thinking about that. You said that size is a component, but I
Mhm.
wonder how much shape influences. I mean, most of the fruit that you chose is pretty uniformly shaped, but I know, for example, in the case of yellow bell, it is kind of bell shaped.
Yeah.
So it's not exactly a circle. And so I'm wondering if you have any other data that could probably um, clarify the effect of shape on the firmness.
Uh, well, as Dr. Juan mentioned, we are working in a nodal analysis to study the relationship between different traits, firmness and external appearance traits, for example, the shape of the fruit. But we are working on that. So we don't have that information yet. But for the calculations, well, most of the trade don't consider that influence of the fruit's shape, except for two that but even those traits are like approximations because the author even say that this is an approximation for kind of convex shape objects. So of course it's not a perfect estimation, but at least in that kind of trait we consider the shape. Yeah, but we actually specifically we haven't studied the effect of the shade fruit in the to the fruit size. Sorry, fruit firmness.
Thanks.
Mhm.
Do we have any other questions for Hector?
I have one more quick one, Amaya.
Go ahead.
Hector, you had one picture where you had a bunch of berries in a container that you were testing for, I don't remember if it was shearing. I just wonder if you found a way or thinking of a way to scale up the phenotyping. You know, phenotyping individual berries can be kind of slow, right? And I'm just wondering if you're thinking of or know of methods where you could speed the process. Right, the Kramer shear cell. Can you do something similar to that with the compression and just
Um,
measure lots of fruits at once?
I think for using a kind of similar approach with the compressions, I'm not really sure if that will work. I I mean I haven't tried that, but according to the company that we from where we get the texture analyzer and the props, I think they have kind of similar system. And however, I think they mentioned something that the effect of the firmness of individual pieces inside in the test could be confounded and not be very accurate as if we test individual fruits. So it could be something that we can explore. But for example, something that is more similar to that idea is the Cranberry cell, and it was like not that terrible, but it was not also the best results. So it's something that we probably could explore because actually the idea for including the Cranberry cell was for having a faster method for texture evaluation because of course sometimes we have thousands of hundreds of samples, but so far the Cranberry cell, for example, hasn't worked very well compared to the additional test like compression test.
Well,
Gotcha.
one caveat to this is that I'm now working with Matt Digman in engineering, and the problem is that sometimes the machines don't have enough force to do the compression for the Kramer shear test. Even the machine that we have that has the capacity to do the Kramer shear test, our fruits are too hard, and depending on how much you use, it just becomes too hard for the machine. So now Matt has a machine that is like an industrial engineering machine. So so we're doing some tests right now. So for VacCAP 2, that's one of our goals is to really test more the Kramer shear test idea. And, we're doing some initial tests. I think that can be part of VacCAP 2 in terms of like preliminary data. I think it's promising, but I'm not sure yet. But definitely firmness is very time consuming if you do it fruit by fruit. So that's one of our intentions is to try to figure out a better way to measure it. And so with Matt, we're looking at 4 or 5 traits related to firmness that might be more high throughput.
Mhm. Also, something that I wanted to
Gotcha.
mention about that is that, for example, for the Cranberry cell, for just one run, we need around 20 fruits, something like that. Thinking that kind of system in a situation where, for example, we have a diversity panel and some genotypes are not able to produce a lot of fruits. So just one measurement is not a good idea, right? Because we won't be able to calculate some statistics like the standard deviation and that kind of things. So in that situation, we will need to run at least a couple of 3 or 5 runs of our climate shear cell, but that will imply that we will use like 100 fruits because for every batch we're gonna use around 20 or something fruits. So for that reason, it's also kind of disadvantage for that kind of methods. But if the ideal scenario will be if we find a similar approach that could use a lower amount of fruits.
Yeah, I think that's a very good point. We're doing some tests with Matt right now about how much, if you do primary shear test, how much in it. Yeah, it takes a lot of fruit. So yeah, with a small plot of 4x4, 5x5, that's definitely a consideration. But for the industry, this could be great.
Yeah, the industry.
All right, any last pressing questions for Hector? Well, if not, well, thank you, Hector, for a great presentation and great discussion of the results of your research. Just to let you know, we will be having another webinar on December 18, Wednesday, December 18. We will have Dr. Jenny Doarca will be presenting on berry portraits, phenotyping of ripening traits with automated image tools and softwares in Cranberry. This is, again, it's going to happen December 18. If you're interested in register, just use the QR code that we have there. I will be sending some emails with the registration link. So with that, thank you very much for attending. We hope to see you in another one of our webinars. And thank you, Hector, for presenting and discussing the results of your research.
Yeah, thank you.
Yep. Thanks, Amaya. Thanks, everybody.
Thank you all.
Thank you. Hey. Muted.